Every cell in your body relies on a master instruction manual. This pristine structure of DNA holds over 3 billion letters of code, and the sequence of those letters dictates exactly how you function, grow, and survive. But that storage system is fragile. Radiation, chemicals, or even normal metabolism can cause a sudden physical break straight across both sides of the ladder, what biologists call a double-strand break. A minor scratch on one side of the DNA is relatively easy to fix because the other side is still intact to read from. A double-strand break is different. When both sides are severed, the physical connection is gone, and the specific letters of code at the side of the break drift apart and are lost in the cellular void. The cell is now on a ticking clock. If it doesn't reconnect those drifting ends, it will die. But because the local information is missing, the cell doesn't immediately know which letters are supposed to bridge the gap. The easiest and fastest response is to just guess. The cell can blindly jam the broken ends back together, hoping for the best. While this keeps the cell alive in the short term, it permanently writes spelling errors, mutations, into the instruction manual. To truly protect the integrity of the code, a simple patch job won't work. The cell needs a mechanism to perfectly reconstruct the exact sequence of missing letters, leaving no room for guesswork. This is where a highly specialized daughter recovery team takes over. You might know them in the context of cancer risk, but their primary job is keeping DNA intact, the BRCA1 and BRCA2 proteins. Think of these proteins as molecular first responders. The moment a severe double-strand break occurs, they race to the scene and latch onto the damaged site, stabilizing the drifting ends. They don't actually act as the physical glue themselves. Instead, BRCA1 and BRCA2 are master coordinators. Once on site, they build a scaffolding and send out chemical signals to summon an entire construction crew of specialized repair enzymes to the break. This crew executes a meticulously organized pathway known as homologous recombination repair. It's a complex term, but it essentially means using an exact matching sequence to safely fix the damage. The arrival of the BRCA team completely changes the cell's trajectory. By securing the site, they block the dangerous guessing mechanism from taking over, and they initiate a sequence designed for perfect accuracy. To rebuild the missing sequence accurately, the BRCA complex needs a reliable reference. It has to search the surrounding cellular environment for a backup drive. This diagram shows the cell solving data loss. Above is the broken sequence, below an exact duplicate template. Proteins stretch the broken ends down, locking on to the matching code. Enzymes then read this healthy template, synthesizing new DNA to perfectly bridge the gap. With the data restored, the newly repaired strand lets go of the template. The remaining tiny nicks are sealed shut, leaving absolutely no physical trace of the original catastrophic break. Because the pathway uses an identical copy as a guide, rather than estimating what belongs in the void, absolute biological integrity is maintained. The restored genetic code is completely flawless. The microscopic crisis is averted. The DNA returns to its normal structural state, allowing the cell to continue its life cycle and divide safely without passing on any corrupted data. This highly coordinated rescue operation happens smoothly, completely out of sight, thousands of times a day across the billions of cells in a healthy human body. Biologists call this sustained state of perfection genomic stability. It is the invisible shield that keeps your tissues functioning correctly and prevents diseases from taking root over your lifetime. Which poses a critical question. What happens if the BRCA proteins themselves carry a mutation, or if they simply vanish, causing this entire backup system to fail? When the cell suffers a double-strand break without BRCA there to secure the site, it panics. To survive the immediate threat of a severed helix, it is forced to regress back to that sloppy method of blindly gluing the ends together. Errors begin to pile up, mutations accumulate with every new break. Without the backup protocol to maintain genomic stability, the cell's code rapidly degrades, creating the exact vulnerability required for cancer to take hold and spread.